Today, we will be talking about advanced data analysis with CLC Cancer Research Workbench and Ingenuity variant analysis. Before we get started, let's review some legal disclaimer. So the QIAGEN products that shown here are intended for molecular biology application. These products are not intended for the diagnosis, prevention, or treatment of a disease. For up-to-date licensing information and product-specific disclaimer, see the respective QIAGEN Kit Handbook or user manual. It can be found at www.qiagen.com/ingenuity or CLC bio. Today's presentation will be divided in a few different sections. Before going within the interpretation tool, I want to start this presentation with discussing about the universal NGS workflow from sample to insight and the integration of the different application and technology that QIAGEN has to offer. Secondly, we will discuss about FastQ to insight, so we will essentially focus the workflow to data analysis and interpretation in the NGS space. After that, we will go deeper into data analysis using the CLC Cancer Research Workbench. So we will discuss about the different functionality and how the Cancer Research Workbench is integrated with the QIAGEN GeneRead DNA-seq target enrichment panel. After talking about data analysis, we will go into the data interpretation using the Ingenuity variant analysis tool. The Ingenuity variant analysis tool leverage a literature platform. So we will start discussing by expert curation and literature knowledge, then review the functionality of Ingenuity variant analysis, and then I will finish the presentation by a little demonstration of the tool itself and wrapped up with a summary. Shown here on my slide, I have the universal NGS workflow from sample to insight. So essentially, as you may have heard already during this seminar series, QIAGEN has a lot of different products for sample preparation. So in the NGS universe, we will do sample isolation, target enrichment, and library construction. When that is done, you then can use any type of sequencing machine to do your NGS run to sequence your actual data. And after that, this is where we are going to focus our day-to-day. We're going to do the data analysis. So how to actually identify the different variants that you may have in your sample and how to do the interpretation. So while QIAGEN bioinformatics carry a lot of different tools, today in this presentation, we will focus on two main tools, which is the CLC Cancer Research Workbench, as well as the Ingenuity variant analysis tool. The first one being the data analysis, the second one being the data interpretation tool. So this is how we go from the sample to actually your sample to providing you valuable insight. What does that mean? If you think about different samples that you may have, you typically will do two main components with a QIAGEN sample preparation tool product. You will do RNA or DNA extraction, library construction, and so forth. With the NGS workflow for RNA, you can go into RNA-seq, or you can go into variant calling. So the CLC Cancer Research Workbench can be used for both workflow. So you can actually generate expression value out of your RNA-seq and use that in a different tool that we call the pathway analysis, where you can look into the different expression of the transcript. With RNA, you can also call the variant and then go into the variant analysis. This is really similar to what we will do today in this demo, but we will really focus on the DNA workflow today, where you actually have DNA extraction, then you do the sequencing of that DNA, and your goal is to find different and identify the different variant in your sample and compare that. It could be, for example, tumor normal, it could be responder, non-responder, and so on. So here we will really focus into this workflow in this presentation today. So we will discuss Cancer Workbench, then variant analysis. So that is what we call the FastQ to insight, so data analysis and interpretation. The solution offered by QIAGEN provides you faster, actionable insight to help you identify, filter, and prioritize those variants. It provides you trusted result, leveraging the most comprehensive knowledge base of its kind, using the knowledge base that we will discuss in variant analysis. Most importantly, it's intuitive and user-friendly. You don't need bioinformatics experience to be able to do and the data analysis and the interpretation. There is ready-to-use workflow for you to do your analysis. It also allow you to do visualization and validation of your result. You can look, for example, if pile up and see all the different reads that you have within the CLC Cancer Workbench. It's flexible. You can customize both in the data analysis and the interpretation, and it's extensible. So you can also leverage and add more and more sample as you go through it. And it's scalable from one sample to multi-sample analysis. So now we have briefly talked about what is the universal workflow where we will focus. We briefly talked about the data analysis and interpretation. And let's go now into what does the CLC Cancer Research Workbench can do for you. What are the main functionality and how it is integrated. And you have different ready-to-use tool if you would like to use the QIAGEN DNA-seq target enrichment panel. You can also use with other different tools. So let's review that. So the QIAGEN Research Workbench is a great tool. As I mentioned, you don't need bioinformatics skill set. It includes comprehensive end-to-end analysis workflow. You don't necessarily need to have all the different background to know when doing which step when you do your data analysis to call your variant. The tool has those workflow ready to use for you. It provides you accurate and trusted results using a fast and easy analysis for whole genome, whole exome, targeted amplicon, whole transcriptome sequencing, ChIP-seq data, and a combination of all these kind of data together as well. So I will briefly show you how you can do that today. It's flexible, customizable. So for those of you that have a deeper bioinformatics background, you can actually customize any of the ready-to-use workflow. You can also build up those workflow, add different steps that you would like. So it's providing you also that flexibility, so it's not a black box. You can actually see all the different workflows and make them for your own specific pipeline. It's allowing you to do validation and visualization of your result. As an example, you can do visualization of variants in protein 3D structure. You can also do Genome Browser. Output, there's QC report for you. And the QC report will also include all the different steps and who does what and which parameters has been used. So it's traceable, trackable when you call your variant. And it works well, as I mentioned, with all the different other QIAGEN products, from the QIAGEN Ingenuity DNA-seq amplicon panel, as well as Ingenuity variant analysis for interpretation of the variant, as I mentioned, and Ingenuity pathway analysis for those of you doing more RNA-seq type of workflow where you are interested in looking the expression value of the different transcript. There's also specific functionality in that tool for human disease data analysis. So, for example, you can do sample genotyping with the list of known variant. You can identify CNV as well as insertion and deletion detection within the tool. How does the tool look like? When you log in the CLC Cancer Workbench, this is how you will see it. As you can notice, right in the middle, you have ready to use information for you, whether you do DNA or RNA. Then you have the different steps. So are you at the data preparation step? Are you at the data analysis step? Are you at the interpretation, or are you trying to do workflow that are together? And every of those links are linking to tutorial that give you step-by-step information how to do the different step within the tool. On the left side panel, you have your data itself. So you have data browser where you can save your data. It could work on a laptop if you have really small type of panel or amplicon that you're trying to analyze and call the variant for. And for our customer that have to do more data intense, computationally intense work, you can also have access to the different server that are access the different servers, so you can run your actual data analysis within the server. And this interface allow you to communicate with that server. If you look below my data browser, you have powerful and user-friendly toolbox. So in the top section, and it's really small due to screen capture, I'm sorry about that, there's the ready-to-use workflow. So preparation of your data, whole genome sequencing. So for those of you that are doing whole genome sequencing, there is the link here. You have whole exome sequencing, target and amplicon sequencing, and whole transcriptome sequencing as well. And if you are someone that would like simply to access different piece of the workflow, you can have access them in the toolbox itself. In the right top corner of your workbench, you also have the different plugin to interact with different other tools, such as variant analysis, such as IPA, such as the Genome panel, and so forth. You can actually access to the workflow building tool, where you can build your own workflow should you choose to do that. And you also have data management, so how to download the different references that you may use. For the QIAGEN Genome panel, we are already providing you all the different references that you can download. You have the human references that will allow you to do your data analysis. If we focus and look a little bit deeper, this is actually how the ready-to-use workflow looks like when you zoom in the section. So you can prepare your data. So you would click on any of those little wizards. So let's say I want to do QIAGEN Genome panel analysis. I would click on this, and there is a wizard that will guide me through everything that I need. So I will have to select which sequencing read do I want to use, what is my panel. So I have a pull down menu where I can select a different panel, and it's already populated for you. Sorry, you don't need to download that by yourself. And then you can also visualize those workflows. So you are, as an example, if you are interested in looking into what is actually all the different steps that the QIAGEN Genome panel analysis is performing for you, you will simply drag and drop that workflow in the right side. So when you are in the application, you select it and you drag it on this white space essentially, and it will give you the different workflow. I don't want to do it here. There's probably 20 to 30 different steps just for this workflow alone, but this gives you an idea of how it's actually built in. So we need first to map the read, then we do local realignment and primer, and so on and so on. So all the different steps are already there. So you can see that workflow. You can change some of the parameters if you want as well. The beauty of the Workbench is also to allow you to see QC reports. So as you run any of those workflows, there is a way for you to actually see what was the minimum of coverage of every of the region and so forth. So you have different type of reporting that allow you to see what is the data analysis looks like, or which region are better and so forth, what is the coverage, where are the primers, and so on. You can also see more information about, who is the user that actually ran this analysis, especially when in instance where you are using a server to connect, you can see who is the user, what is the parameters that was used to call those variant, the minimum coverage, what is the cutoff that was used, have you asked to create a report, was comment entered, so did the user that generated this data analysis, did they enter comment, and so forth. So you can really track every step that has been done, and you can come back in a year from now, and then you have all that as your fingertip. And then when you actually look at the result itself, you have that Genome Browser view that allow you here as an example to see what is the different variant, what is the pileup, so what are the read, what is my actual human sequences. So there is a Genome Browser view where you can see my genome sequences. I can see where my primers are. This is a track for my primers. I can see here the different read that I have. Here's my variant, and you can see the screen capture is small, but you can also align any other type of annotation that you may have, including genome tracks that you can import. And as you click on a variant, you will have a window that will allow you to see deeper, which chromosome, which region, what type of variation that is, what is the references, and so forth. So you have a detailed information. And if you mouse over, you actually have a lot of information. So if I mouse over one row as an example here, I can see more information about the coverage, forward, reverse, balance, and so forth. So you have all that information that is available for you. From there, that's when you can also start and push this data into the different other tool, or export that as a variant calling file if you are interested. So if we zoom in a little bit more into that rich variant level annotation and dynamic, you can see here what I was illustrating. So you have your human references. You can see where your variant are. The color means something. You have your amino acid here. What amino acid here? Here I can see that this is actually changing. This particular variant is changing from Q to R, and then you can see you read which one are forward and reverse and so forth. And you can drill down into the information, chromosome 17, this is the region, it's a single variation, T to C, frequency, coding region, amino acid, DB SNP, and so on. So you can really see and browse into that information. Now, a little bit more about what does the QIAGEN GeneRead panel allow you to do. It's allowing you, as I said, to identify and annotate those single variation, small and large insertion and deletion. So with your small and large indel, this workflow will do that for you. It will analyze using the standard panel, focus on a specific set of gene, using all the target, primer, and so forth region of interest. It's already included into the Workbench. It will accurately remove the different primer after the mapping to provide you more accurate variant frequency and genotype information. It will automatically remove the read with primer dimer issue, so to improve the specificity in the variant calling. And if you need more information, it might take a little bit longer to do this, but there is QIAGEN GeneRead panel, so we have help documentation only talking about the specific plugin here that is already included. This was just a brief overview of what the CLC Cancer Workbench can do. Now from here, when you are actually in this window, if you right click, and unfortunately I don't have a screen capture here, you can actually right click into this result and send using a different plugin. You can send directly the data into the data interpretation tool, which is Ingenuity Variant Analysis. So before going into that, what is Ingenuity Analysis and how can you leverage a tool to do interpretation of variant? Well, Ingenuity Variant Analysis is using comprehensive human disease variant content. So what that means is we're using more than 15 years of experience of manually created information, and we are having a really big effort into creating all the human disease finding information. As of the end of last year, I believe this number is relevant for November, we have over 2 million different finding. And if you look into that, talk about the specific variant, and if you look only into the Ingenuity specific created finding at that time, we were over 600,000 different manually created finding. What does that mean? What is a finding? How can we get to that information? So what we actually do at QIAGEN Bioinformatics is we have a set of scientists that would follow a strict protocol, and depending of the different finding, whether it's a gene finding, whether it's a variant finding or a clinical finding or the different other tool that we have, they follow really strict guideline and protocol and read literature that are interesting, from basic research sometimes for gene finding to clinical report for variant type of finding causing disease. And they will do what we call content acquisition, follow the protocol, enter all these interaction. Could be gene A is causing disease, or variant W in protein Z is providing a gain of function or loss of function and so on. So they will enter all that information. We have a team that will manually QC those findings, and everything will be connected together using our QIAGEN Ingenuity ontology, where we will look into from the patient phenotype to the sequence mechanism. So we will connect all these different pieces of evidence that we have on disease, evidence that we have on patient phenotype, cellular mechanism, molecular mechanism, to the actual sequence mechanism that could happen on the DNA or the RNA type of information. We also do on pathway modeling, importation of different public database, and you will see that when I will go directly within the tool, the type of database that we are importing into the tool. And this is, what is behind variant analysis? What is actually the platform that allow variant analysis to help you identify those causal variants? And the way we will do that is you will, in the cancer workbench, sequence, you do your sequence, you align your variant, you call your variant. So the cancer workbench would do that. And variant analysis will take the information at this step. We'll take the variant call file, either by export, either by directly link from, as an example, the CLC Cancer Workbench, or the tool that you've been looking at in the webinar series last week, the online tool that we have also at QIAGEN. And then essentially these variant call file will be uploaded into variant analysis. Then you can combine them depending of the different type of analysis that you may have. Maybe today you have a trio analysis to do. Maybe tomorrow you have to look into 20 different tumor versus 20 different normal tumor match sample as an example. Maybe you're doing big cohort and you're looking into responder, non-responder, treatment after disease, and which variant may differentiate the responder to the non-responder. So you can combine and you can mix and match these different samples together to generate an analysis. And there is a really easy to use workflow wizard that will allow you to do that. And the goal of the analysis is, of course, to annotate those variants, also allowing you to indirectly filter those variants down. So if for whole genome, as an example, many of our customers may have 3, 4, 5 million in every whole genome. In Amplicon, of course, it's smaller, but you still have a lot of different variants. Which one or which 10 or which 20 are the most relevant for the question that you're having? So variant analysis will allow you to filtering this down using different steps, and the step will be different based on the type of analysis that you selected, but overall we look into confidence. Can we filter based on the quality, the read depth, and so forth? Can we filter based on who, removing those variants that are commonly occurring into the population in general? Then go deeper and deeper into the biological interpretation, keeping only, for example, variants that are causing loss of function or gain of function, or any criteria that may be beneficial for your analysis. For pedigree or trio analysis, filtering based on that Mendelian inheritance association, and finally making biological context out of it. And what that does, it's allowing you to go above and beyond what is known. So maybe today, we know in the literature, because of the curation, we know that a variant in gene C will cause a given disease. But maybe the variant that you have in your file or in your sample are in gene B or gene A. Because of all the other information that we have in the knowledge base, we can run analytics and allow you to say, well, your variant that is in gene A causing a loss of function may actually impact gene B, because there's a loss of function of this gene. Therefore, all that signaling cascade may or may not be relevant to a disease or particular molecular function that you are interested in. So it's allowing you to link the variant, the biology in the knowledge base, to go above and beyond what is to be published in the literature. And finally, variant analysis also allow you to share and publish different sample or your analyses as well. So you can share with your colleague, you can publish using variant analysis. And the key challenges that are addressed by this tool is to help you prioritize and filter the false positive, help you with the interpretation bottleneck, and maintaining and synthesizing the relevant content and information on your variant. And variant analysis is a web-based application, secure web-based platform for interpreting called human genome or amplicon and so forth, so human type of sample. As I mentioned a few times, again, similar to the cancer workbench, it's flexible. You can annotate flexibly, you can change the different parameters if you choose to do so. So it's allowing you to shortlist the candidate variant gene or gene set that are relevant for you, for your particular question that you have in mind. It also have statistically robust method to interpret matched tumor, probe and kindred, big cohort, and so forth. It's shareable to allow you to do collaborative discovery with your colleague. And it does have deep functional knowledge with rigorous curation of published finding, a well-structured ontology was been developing for more than 15 years in interaction modeling, and there's also HGMB that is integrated within the variant if you're interested into the germline variation. So how does the tool look like when you actually are looking into an analysis? So before going into my live demo, let's look at how it looks like. So here, it is an analysis that I have took screen capture on. This is a cohort in this case. This is a really large cohort. So I will have on my left side of my screen the filtering cascade. You know, that will be divided in, you know, how many variant total I have for all the sample in this analysis corresponding to how many gene. And every step of the cascade, I'm losing variant based on the criteria that are happening into every of those filtering cascade. And as a consequences, number of gene go down and down. This particular example here is actually a data that you can see. It's published in every customer, regardless if you have paid sample invariant analysis, you can actually look into this one. And it's a big cohort using the PGP example where we were, in this case, trying to find variant that could be potentially relevant for myopia. And you can see the different statistical association that was performed and different tests that was done, where at the end we identified potentially at the time of the screen capture around 100 variant corresponding to 13 gene. And on the right side of my screen, this is what we see. We see, you know, the chromosome, the position, what type of variation that is, single variation, insertion, deletion, substitution, and so on. Where that is in the gene, is it in the exonic region, is it in the intronic region, and so forth. Splice site, what is the corresponding gene symbol, the protein variant, and then you have many other columns that you can add to explore the data. And the goal of this filtering cascade is you're keeping the one that are relevant for you. So typically most workflow will start with filtering the quality of the variant call, removing the usual suspect, those variants that are happening always because maybe a protein or a gene is too long and so forth. Then you could do assessment. In this case, we do assessment around pathogenicity. Is this likely pathogenic, not likely, benign, gain of function, loss of function, and so forth. Frequency of the variant in LT population is also another step that you can use as filtering. You, in the case of large cohort like this, you could identify the variant based on basic association tests, comparing case and control sample. There's also quantitative trait and so forth, tests that you can perform using that that filtering that filter. And then you find, using the causal network, you identify the biology that is relevant for your variant, essentially. And this is just an example of this one, which is myopia, where we went above and beyond and said, well, we want to identify variant and gene that are known to affect either myopia, eye development, eye morphology, lens degeneration, and so forth. And you can add as much text as you want here in order to tailor that to what is relevant for you. And you can do affect, directly affect, inhibit particular process or diseases and so forth, upstream or downstream of the criteria that you are entering in here. So what I will do now, I will go within variant analysis. So this is the tool, as I mentioned, it's a secure web platform tool. When we log in, if you do have analysis, you will typically log in this section here where you will have all your different analysis. And we can see here, as I click on any of the analysis, I see, what is the name, description, if someone shared it with me. So I shared it with myself, when it was created, how many samples. So this one I have 1 out of 1. I have 1 cases, 1 control. This one I have 4 cases, 0 control, and so forth. So you see some data. You see whether or not your analysis is active or inactive. And then you have information, what are the filters that are used, what are the samples that are used in this particular analysis. So this will give you some information about your different analysis. If you want to open an analysis by itself, you will simply double click or click open, and then it will open your analysis. Before I go there, the way to actually run an analysis, if you use the Ingenuity variant analysis plugin from the Cancer Workbench, which will be in your ready-to-use workflow in the plugin section. If we look here, you could see that I actually have a variant analysis plugin, so I can send directly my tool, my sample and variant analysis, and it will run the analysis for you. Similarly, when you are using the web platform tool that was discussed in the webinar of last week, it will also run the analysis for you. If you want to run the analysis by yourself, you can also go into my sample section, either upload your sample individually or using iVolume Uploader, but upload your sample into your toolbox. Pick any of the samples that are relevant for your particular analysis and click Analyze, and you will get through a wizard that will ask you a few different questions, such as genetic disease. Are you interested in studying a hereditary disease or syndrome and seeking the causal variant? Are you looking to identify normal and tumor? What is identify the cancer driver variant? Stratification studies such as responder, non-responder, and you're looking for variant that distinguish the two. Personal genome, so maybe you have only one file and you want to look for those variants that are linked to known disease or phenotypic association. You want to actually create it yourself, or you are a returning customer and you want to use setting that you have been used in a different analysis. So it allow you, as you build your pipeline, as an example, if you always do responder, non-responder, and always want the same filter to be applied with the different experiment that you're doing, you can also select this option and actually look into your analysis and always use the same analysis. And you guide through, identify case control, sample condition, and so forth. The length of the analysis, running the analysis, can be a few seconds, a few minutes to, typically if you're talking about 200 whole genome and things like that, it could go to a few hours, 200, 300 whole genome. But it's nice because it's allowing you to essentially do that interpretation fairly quickly on the web, so it's securely transferred to a secure server. And when your analysis is ready and loading, this is how you see. So here we see, here we see this one analysis where I've used a GeneRead cancer panel. And this is just an example of what can be done. And as you can see on the left side, I have my filtering cascade. So it's dynamic. I can see the number of variants. So of course, we're not talking about whole genome here. So I started with 200 variant. corresponding to 34 gene, and then I apply a different step of filtering. For every of those steps, you can see what they are. So you can click on this little paper sheet that will allow you to see what are the criteria. So quality for confidence, upstream pipeline, read depth, most exonic variable, region or variable gene, and so forth. So every of those filter, you can actually go and change any of the parameters. So I can go and say, okay, I want to change this because I want to exclude variant given a certain criteria, and change any of the criteria, and you can go apply and change any of the filter. I'm not going to do it at this time, but it's simply to say that any of those parameters can be changed, can be moved up and down. So maybe you're like, well, I really want to do this step before that other step. So you simply use the little arrow to move the different filter criteria, and invariant analysis will just rerun the analysis for you automatically. If you need to access the online help to see what can be done, what does rarity or this filter mean, and so forth, you click on the little eye and it will link you to online page where for most of the filter you will have a video as well as description of what this filter is about. And it's as I said, given the wizard that you will be using, that filtering cascade will be different. You can always add different steps. So here, as we can see pharmacogenomics or pharmacogenetics information. And this was added. I actually went actively and said, I want add filter, and I wanted to focus on a particular drug of interest. So I can select any of the filtering step and add more filter to tailor my analysis even deeper. Okay, so in this example, the filter that was added is I wanted any drug, every variant where a drug has been found to cause response, toxicity, or is an established target, and so forth. On the right side of the screen, you have your results. You have the different chromosome, where is the position, and so on, as I discussed. And for any of the variant, when you click on the row, you have at your fingertip all the information, so you know what they are. You have a visual diagram, case and control. Here I have only case in this analysis, so I have only tumor in this case. I can see that 2 of my sample have a nomozygote variation, and then 2 of my sample have a dash, means that there is no variation for these 2 other sample. And the meaning of that legend is actually below the filtering cascade. So the blue will correspond to a loss of function. The black will correspond to something we call normal, or meaning that not necessarily that the variant is normal, but we can also have no information about whether this particular variation has been shown to cause a loss or gain of function. So in this case, we're not sure, so we're putting a black coloring. And the orange means gain of function. And the different square or double square and so forth are relevant to the genotype. So heterozygote variant, ambiguous, homozygote, copy number gain, and so forth. I'm sorry. So that's what we see here. We have double shape for homozygote variant, blue being a loss of function in 2 of my sample out of 4. The translation impact is a stop gain. For this one, it could be in frame, missense, and so forth. So you can see all that information. You have, if you think it's a CIF information, polyphen. So all of this is already within the tool. You don't need to go and look into CIF website and polyphen website and so forth. It's already there for you. And you have many, many other different columns that you can add. And when you click on any row, like I have here in blue, you actually see the information on the right side, completely on the right of your screen. So you can see a view section where you can actually access directly the variant finding, the pharmacogenetics information we have for that given, what is the gene. If you click on the gene name, so everything in blue is an hyperlink. So if I click on the gene name, it will be linked to the gene information. So all the information that we have around this gene. It will also, while this is loading, it will come back. It will also, if there is a DB SNP information, you will see that. What is the cytoband? Cosmic ID. So when this variant has been shown in Cosmic, you will see that. You can validate that Cosmic ID, see the position. You can go in IGV, in UGV, and so forth. Let me make this a little bit bigger. UCSC Genome Browser. When the variant is in the HGMD databases, you are able to see it. So you are able to see HGMD. What is the tag for HGMD? If you do have a subscription, it will link you directly to the different databases. If you don't have, you will see the public information. If you have a HGMD subscription for professional HGMD subscription, you will also be able to go and see the deeper information. You also have a classification. So we are classifying every of those variants based on some criteria. And if you click on the result, pathogenicity, here we will populate for you the evidence of information that we have. Why do we say that this variant is potentially pathogenic? It's based on an algorithm that is using classified computed classified information based on the draft of ACMG guideline 4, and then you have the different disease that is 4, and then you have the evidence of pathogenicity and against pathogenicity. And for every of your variant, you will see that. And the beauty of the online tool is if I come back in 6 months from now, if there is new literature evidences, this will just be enriched. So I will see more and more evidences as an example. Or if there is a publication that is completely reverting that information, I will also see that. Then you have many other information that you can get for every of your variant. And from there is also a concept of gene, group and complexes, which pathway those variants are involved in, which pathway are perturbed, what are the processes that are affected. And we do different statistical measures to tell you, well, this pathway may be involved, this other pathway. So if you look into colorectal cancer metastasis signaling pathway, you actually have a view where you can see where your variant are. They're color-coded using the same code. So blue will be your loss of function, orange will be your gain of function, and you can see which gene they are. You can highlight which of those gene or drug target if you are interested in looking into a variant that may perturb the effect of some drug and so forth. So this is a lot of information, so I will pause here for the demo. The goal was really to give you an overview of what you can do in interpretation. So interpretation will be the variant analysis tool and data analysis, which means from your sequence to calling the variant. This is done into the cancer workbench. So if I go back to my PowerPoint presentation, in summary, we looked into different variation. I showed you where, a little bit about the different interface that you have access, the type of information, the assessment, and so forth. When you click on any of those links here at the top, variant finding or pharmacogenetics or haploinsufficiency, you actually have all the finding, all the literature evidences. And here we're looking at haploinsufficiency, where I can see all the different findings that are talking about haploinsufficiency for the variant at this chromosome at that position, which is a single variation and change to a T. And it's dynamic. Same thing with the variant finding. So here we're looking into, at the time of the screen capture, what was the finding we had? Well, we had variant finding information from manual created, from Cosmic, and so on. So you see all the different variant finding when you click on that variant finding link. This is just, again, another example with a different gene, the APC gene from that analysis I was showing you. And the pathway allow you to go, above and beyond, look at to how actually all those different variants may affect a particular pathway. So you can draw a conclusion about interpretation as well. So, what we talked today is really the first FASTQ to Insight solution from QIAGEN. And it's, I haven't demoed it during this presentation, but you can actually go back and forth between Ingenuity variant analysis, you could identify a variant and go back into your cancer workbench and say, I want to see the pileup of this. I want to see actually all the data. So you can go back and forth between the workbenches and variant analysis to allow you to identify the variant, maybe tailor your analysis, and then come back into the cancer workbench. Oops, I'm sorry. For some reason, my computer is slower. In the way you do that integration is, as I showed you in summary, essentially you identify the variant using those workflow. You can use the plugin to send the data into variant analysis. You can also create a workflow and add that, so it's automatically doing it from the beginning to the end and sending it in variant analysis. You can see the result within variant analysis by clicking on those links, and you can come back in variant analysis to update everything that you did. Let's say you change an analysis, you can come back in your workbench and click variant analysis update and update that information and see in your Genome Browser, this was the variant before I went into variant analysis, and this is after I went into variant analysis, because maybe you added another filter that was not there in the original analysis, and now you actually, if we look into this screen capture, what I did in variant analysis removed the variant, so I lost my variant. So it's a lot of information from this presentation, but I hope it gave you an idea of what you can do into the bioinformatics product using the cancer workbench and the variant analysis. And I want to end the presentation with some, and I'm not going to read it, but some quote from our customer, both for the different product about cancer workbench. It's really time. In data analysis that was taking weeks or now taking a few days. We have a support team that is there to help you with the different products.